All right, so today we're going to talk about pancreatitis and how it can lead to pancreatic cancer. A lot of times people have pancreatitis. It does not lead to pancreatic cancer, but it does make pancreatic cancer more prevalent. I'm going to get into the statistics a little bit more than normal about what leads to a disease, because this is just a sad story currently where we're very bad at detecting it, and once you have it, it's not really that treatable. I would expect this story, pancreatic cancer in general is kind of hard to predict, so I'm much less sure about this than in 10 years we'll all be sequenced, but I would expect this story to be a lot better in 20 years, okay? And I'll speak to you about that. So we're going to talk about what's happening when you're ingesting these different types of food. When does that go wrong? Carbs, protein, and fats. We'll get kind of in the cell how calcium's involved. Also, long ago in Foundations, I talked to you about how vesicles were releasing contents, and I told you I was kind of giving you the undergrad simplified version, pulling the wool over your eyes, and we would get into the detailed version later. Now we're going to get into that detailed version where I think of it like a space shuttle docking to something. We'll talk about the ramifications of that, and yet again, we'll talk about how alcohol is bad for you. All right, let me go back. Let's see if this will open. I was just going to show the notes for Panopto's sake. Here is the table of contents. As always, read at your leisure. Okay. So we're going to be talking about zymogens, pre-active enzymes, the releasing of bile. We'll get back into cystic fibrosis because they have issues with pancreatitis. We'll talk about when things go awry and you can't digest oil if you have oily stools. And we'll bring up the omega fats one more time. So this is a few years old now, 2017 data. I compiled this to show you the order of magnitude difference. First, let's look at the left side of the screen. So here we're looking at the estimated number of new cases. This is for pancreatic cancer, okay? And so for men, the new cases of pancreatic cancer for the pancreas is just 2%, okay? It's a low amount of the cancers. For females, it's also 2%. When you look at the estimated deaths, for males, it's 5% from pancreas, and for females, it's 6%, okay? So it's less likely, it's one of the less likely cancers, but it's a killer. I didn't feel like that really drove home how big a difference it was, so I put the 2017 numbers in percentages to compare. So here is the number of diagnoses per year, okay? 53,000. If you compare that to the population, okay, that's 0.017%, very low. A small percent of the population is diagnosed with this, right? But if you look at the number that die per year, 43,000 die per year, and if you compare that to the number of deaths per year, it's 1.6% of the deaths. So there is a 100-fold difference, okay? So this is kind of the silent thing that we can't detect, and then it's too late and you're dead, and we'll talk about why that is, and we'll talk about future ways to potentially solve that. But unlike, you've already had the dementia stuff from me, unlike the dementias where people are trying, like for Alzheimer's, to detect it earlier to treat Alzheimer's, even there, when you roll back and try to detect earlier, probably the brain's already being degraded. Here, it should have much more efficacy. If you can detect it earlier, you're going to get the cancer before it's spread. That's the problem. And so then you can cut it out, you can do chemo, you can kill it at the source. So whenever we tell you a horrible story of like, this is horrible, there's a lot of room for improvement. That's the silver lining to this story, right? Nowhere to go but up. Okay, so pancreatitis can lead to pancreatic cancer. Let's talk about the different types of pancreatitis. There's acute pancreatitis, and those can result in gallstones. And so this number in parentheses show kind of the magnitude of that, 15 of those per 100,000 people per year, okay? Then you can have acute pancreatitis that's non-gallstone. It's 2/3 of the pie, so 25 there, okay? In the acute pancreatitis section of the pie, you can end up getting recurrent acute pancreatitis. That's 10 of the 100,000. And from those, you can get chronic pancreatitis, and the chronic pancreatitis can lead to pancreatic cancer. Okay, so this is kind of the flow chart of how concerned you would be about a patient. Okay, do they have acute pancreatitis with gallstones? Okay, we're treating the gallstone, but we're not as worried about cancer. And once they get down to recurrent acute pancreatitis or chronic pancreatitis, then you have to start saying, how can you intervene as a patient to prevent your risk of pancreatic cancer? Not to mention, pancreatitis isn't great either, right? Okay, so what is contributing to pancreatitis? The main thing that you will hear, so you'll probably see that on the boards, would be alcohol, okay? I'm gonna add smoking to that, and I'll show you the data. Smoking and alcohol both contribute to pancreatitis. So let's look at this bar chart on top. This is for acute pancreatitis, okay? This two-thirds pie chart of the big pie. And in purple, we have what percent is due to alcohol. In yellow, what percent have gallstones. And then in blue, what percent is idiopathic? We don't know what it is. And as you go up, let's look at males and then females. As you go up into kind of middle age for males, you see about half of it ends up being due to alcohol, okay? And in females, it's a lesser amount, maybe a third in midlife, and then that drops off later in life, okay? Now, if you look down at chronic pancreatitis, so a more severe version that can lead to pancreatic cancer, alcohol kind of plays a bigger role and for longer in life, okay? Now, I'll show you data in a second that, and I'm kind of reading between the lines of two data sets here. The data I'm about to show you, I would suggest that some of this purple idiopathic bar chart is due to smoking. I think that's pretty clear. I just don't know what fraction of that purple is due to that, okay? Everyone clear on what I've said so far? Okay. So again, this is taking a bunch of data and putting it in kind of a heat map table format so you can get at what is the magnitude of this importance. So first, let's look at what these words mean, okay? We're gonna be talking about alcohol and smoking here. So what is a type of drinker? An abstainer is 0 drinks a day, okay? A light drinker is, I'm just gonna go through drinks a day. This other column is drinks per week. A light drinker is half a drink a day. A moderate drinker, the definition starts to toggle between men and women, and that's basically because of body mass. So for women, a moderate drinker is 1 drink a day, and for males, it's 2 drinks a day. And the take home that I would give you is kind of anything from moderate down is okay, okay? Moderate probably has a little bit of a hit, but if you kind of think of a graph of the magnitude of effect, you're very on the low end of starting to see bad stuff, okay? Once you get to heavy drinker, which is 1 to 5 drinks a day for women and 2 to 5 drinks a day for men, it starts being worse, and then very heavy is 5 or more, okay? And so in this tables to the left that I'm gonna show you, they have this study grouped things into heavy plus, and so that is heavy and very heavy combined, okay? So big take home there, 1 to 2 drinks a day is not bad, 1 if you're a woman, 2 if you're a man, and really I wouldn't even go mainly by sex there, I'd go by your body mass. So a woman and a man that both weigh 200 pounds can probably have 2 drinks a day, and a woman and a man that both weigh 130 pounds can probably have 1 drink a day, okay? So now let's look at the effect of combining smoking and alcohol, okay? On the, going across the top, we're gonna see drinking categories, and going down, we're gonna see smoking, okay? And they've all been normalized to never smoking, right? They're all set to 1. And this table is all odds ratio. So if you see a 2.2, that would mean 2.2 times as many chances. So a 220% increased chance, okay? So if you, and the top, this is the same, it's representing the same data, but the top table shows pack years. You guys are familiar with pack years now, is that correct? Yes or no? Okay, right. And the bottom is pack days. I'm just gonna kind of work through pack days because it's a little more intuitive to me, how many packs you're smoking a day, but there is a take home that pack years is important in a longer term 5-year thing, because maybe you used to smoke a ton and now you're not, but you still have a hit from that, and you can get that in pack years. So in packs per day, if you are a never drinker, okay, you're set at 1, and as you start smoking more, if you get to just one, let's say 1 pack a day, and you still don't drink at all, your risk of chronic pancreatitis, which can go on to lead to pancreatic cancer, is twofold. Okay, so when you talk to a patient about this, you could say your odds ratio is 2, or you could say you have twice as much chance. I think when things are very serious, it might get the effect through more if you represented it in percent, because it is as true to say you have a 200% chance of getting pancreatitis compared to a normal person. If you go over 1 pack a day, it didn't really change that much. It went down, and that's probably, I would argue probably, well, I don't know. That shouldn't have made sense. It should have kept going up. In the other categories, you see that it does go up. I'll mention why. Do you remember my story about why the data is weird for abstainers of alcohol? I mentioned this a while ago. So it turns out maybe a year ago or 2 years ago, someone looked at all these old studies that looked at, and this is across the board, looking at alcoholism with multiple things. If you look at never drinkers and then light to moderate and then heavy, light to moderate seemed to be the best. And people were like, oh, Mediterranean diet, you need to have a cup of wine, right? This is that thing that we've heard, a cup of wine at dinner is good for you, heart protection, blah, blah, blah. Well, it turns out that a lot of that data is probably because in the abstainer group, they had prior alcoholics that had become abstainers. And so they had a hit. So they looked worse than the people that for their lifetime had only had 1 drink a day, because someone had drank, a massive amount and then become an abstainer. So that data all became suspect. So the trends all seem to make sense when you look from light to moderate to heavy, and then sometimes it goes the opposite way in abstainer, and I would put an asterisk next to that and say, I would guess that generally no alcohol is the best, and it just goes up, okay? So anyway, so if we continue down here, if you're a light drinker, your odds of getting pancreatitis with 1 pack a day is 1.3, and then they almost triple when you become a heavy smoker, from 1 pack a day to over 1 pack a day, okay? These stars are statistically significant. If you're a moderate drinker, you have a twofold at 1 pack a day, and then again, it goes up to 3, you see maxed out. When you get too heavy, things start to get very bad. So if you're a heavy drinker, okay, and you are 1 pack a day or under, you have I would say this as 450% chance of getting pancreatic cancer, or over 4-fold as likely of getting pancreatitis, chronic pancreatitis. And then when you go to heavy, it becomes 6-fold. This is pretty, I mean, when you look at odds ratios for diseases, a lot of times you'll see like 1.4, which is a 40% increase. To see a 600% increase for something that then leads to cancer is very big, okay? Now, I'll just point out that if you look at pack years, so the long-term trend, it is even worse. If you are a heavy drinker and you have over 35 pack years, your odds ratio are 1,300% of getting chronic pancreatitis. So this sounds horrible, but this goes back to, I don't think I showed it to you guys. Email me if you want to see a table I made on lifestyle interventions and what you can do, and they're rank ordered, like for all things. And the number one thing you can do is not smoke. Not smoking is like, if you want to save lives in America, that's the one to hit. Don't smoke. And anyway, for chronic pancreatitis, the combination of the alcohol and smoking is important. On the boards, even despite what I'm showing here, you will probably, the triggers in the stem will be alcohol for pancreatitis, okay? Any questions about any of that? Yeah.
Has there been any research on e-cigarettes and pancreatitis done yet?
The question was, has there been any research on e-cigarettes and pancreatitis? Not that I know of. It might have been done. It's pretty soon to have been done, but it might have been done. I don't know the answer to that. Yeah, I would guess the effect is not as large if there, but I don't know the data. Yep. E-cigarettes, that's a interesting story we can talk about offline if you want. Okay, so When you're talking about the pancreas, you can be talking about endocrine or exocrine. This lecture, we're just going to be talking about exocrine, and we're going to be looking at these acinar, stellate, and ductal cells, okay? And if you look over here, the acinar cells, these blue ones, can be thought of as the factories that are pumping out your nanomachines, right? So they're pumping out zymogens, these enzymes that are going to go and digest stuff. So if you have a meal, your acinar cells pump something out, and they're like, okay, let's go handle that food. Your ductal cells can be thought of as the highway, right? They're the duct that those are going to travel down. And then your stellate cells are kind of these supporting cells, okay? And so we're going to talk about how, when you make your zymogens, how they go here, dock, and get released. What can go wrong? How do they get activated? What goes wrong if they get activated too early? And then there's examples of they can even be released backwards the wrong way. And so you can degrade tissue this way and cause necrosis. Okay, and so if you just kind of think about it conceptually as factory, highway, and supporting cells, it kind of makes it obvious when things would go bad. All right, this is as good a artistic rendition as I can have. So here we have the stomach. As you eat food, you come down and you have I cells and S cells that detect the food, okay? Let's start with S cells. S cells secrete secretin, S and S, okay? So they detect here, but the secretin, in your view, is secreted to the left. It is secreted into the blood, okay? And then the I cells release CCK, also secreted into the blood. And what do those do? The secretin increases bicarbonate. So you have this very low pH in the stomach, and you're going to want to bring that pH up, okay? So you're releasing bicarbonate. And then the CCK releases bile and pancreatic enzymes, right? And so the bile is helping to emulsify the fats, and the pancreatic enzymes are breaking stuff down. This table here shows things that you can measure for diseases. So for acute pancreatitis, you can measure lipase and amylase, okay? This table continues off the screen. I'm not testing on any of that, but I just showed you other diseases and what you can measure for board prep. Okay. All right, so this table down here, first of all, as with all my lectures, any chemical structure you don't need to know. So this whole thing up here is an FYI. I'm just showing you that the different enzymes that break apart amino acids break them at different consensus sequences. You don't need to know that, not board testable. This table down here I made in a readable format across, okay? So this proenzyme is cleaved by this, creating the active enzyme, which cleaves this. So chemotrypsinogen is cleaved by trypsin, creating the active enzyme chemotrypsin, which cleaves protein, okay? And so let's start with trypsin, or trypsinogen, because you can think of that as the spark that lights everything off, okay? So you have trypsinogen, which is a zymogen. It is an inactive enzyme, but it can be cleaved and become active, right? Now, some of the minutia that we'll get into and common questions I get, I think that if you have trypsinogen just by itself and you left it there for a while, in some amount of time, it could auto-cleave itself and become active, okay? So you have a protein sitting in the duct, I'll show you later. So as the zymogens come by, you have this protein enteropeptidase, okay? And it's sitting there, and as trypsinogen comes past it, enteropeptidase cleaves it and makes trypsin. Trypsin then can activate a bunch of proteins. One, it can act on trypsinogen, so it can act on its own zymogen and activate it. So if you had a vat of trypsinogen and you put in one trypsin, all the trypsinogen would become trypsin. Let's say that fast 5 times, okay? So anyway, trypsin can auto-activate trypsinogen. It can also activate chemotrypsinogen, making chemotrypsin. It can activate proelastase, making elastase, and it can activate the procarboxypeptidases A and B, making carboxypeptidases A and B, okay? So all of these top ones go on to cleave protein, okay? Then we have lipase and procolipase, which are involved with cleaving lipids, triacylglycerides, and then amylase is involved in degrading polysaccharides. I'll point out that procolipase is, you can kind of think of it as a modifier of lipase, right? It increases lipase activity. So procolipase is cleaved by trypsin, creating colipase. Colipase binds lipase, and it basically allows it to metabolize the fats better. And the reason you need this is this goes back to like this generic thing of you are ingesting various food sources. We're basically bags of water. And when you ingest a fat, the fat aggregates by itself because it doesn't want to need to be in water. So what do you do with that? You have bile, you emulsify it, and you break it apart so you can act on it, right? Well, so now you have some kind of oily bile emulsified thing that you can act on, and something's going in there to act on the lipids. That thing has to kind of be hydrophobic too, because it has to get into this, okay? And so the take home there is lipase can actually be put in this emulsified layer too much that it doesn't work, and procolipase keeps it kind of near the membrane, working on things as it goes. It just thinks about it, kind of think about it like a chaperone. I don't know if it's really quite that, but it basically increases the activity of lipase. And we'll get into that because you can actually have problems. There's obvious problems if you have no bile, but you can have problems if you have too much, if you're emulsifying too much, and colipase can help with that. All right, so now we're looking at, again, just those bottom parts of the table that I just showed you. So you have your triacylglycerides being broken down into fatty acids. And so you don't need to really know these breakpoints. I'm just showing you with the lightning bolt where it gets chopped. But the cholesterol ester is converted into cholesterol, and bile salts help that, right? So bile salts help get at that. And then if you're looking at phosphatidylcholine here, bile salts, if you have a lot of them, can actually inhibit this, and then colipase helps it, okay? So it just gets down to that table I was showing you before. Any questions about that? All right, so let's look at the right graph here to kind of give you the big picture, and then I'll walk through those words. So for hereditary pancreatitis, there is a huge lag between when things probably start going bad and then when you have exocrine or endocrine failure, about a 30-year lag, okay? And so you have some first symptom that we're not really detecting. So basically there is the possibility that we could find things out earlier before there's massive failures to take home there, okay? So let's get through the risk factors. Obviously things are getting worse as you age. Diabetes, alcohol, and smoking, we already talked about the latter two. If you are over 60 years old and have adenoma in the colon, there's a 40% increased chance, okay? If you have a colonoscopy that shows more than one or more adenoma, depending on your sex, you're either 15 or 25% more likely to go on and get pancreatic cancer, 25% for men, bigger hit. And if you have a 60-year-old first-degree relative that has had a colon adenoma, then you're at 2.5-fold increased risk of colon cancer. So this is all family history type stuff you can get. Okay. And hereditary pancreatitis increases the risk of pancreatic cancer. There are some genetic effects in the patient that you can see. These are rarer, right? Most of this is caused by the environment, smoking and drinking and age. But there's this gene, PRSS, and there's a version 1 and 2, and they're cationic trypsinogens, or one's cationic, one's anionic. But the PRSS1 is a gain of function, okay? And so this is opposite than the normal thing I told you. Normally I tell you if you have an enzyme, right, and a trypsinogen is an enzyme. If you have 2 of them and you lose one, you're fine because you still have the active one, right? And normally if you have 2 and you have more of them, you're fine. You just have more of the active one. But here, it's bad if you have more of the active because you're degrading things too early, right? And so if you have this PRSS1 gain of function mutation, then it creates a zymogen cascade where you're activating the enzymes too early, and so basically you can start having tissue failure or necrosis where you're breaking down things in the duct before you get where you're supposed to go. And that's 65% of those genetic factors. 30%, the other third, are from PRSS2, and that mutation is a loss of function, so that is a beneficial thing, hence the green. Because there you're kind of tamping down the match. You're being like, okay, let's not go crazy digesting stuff, and so then it doesn't digest stuff in the duct on the way. Okay, and so those have an autosomal dominant inheritance pattern, which should make sense. Okay, so how do we treat pancreatic cancer? First of all, I've told you that it's super deadly. So I'm going to tell you how it can be treated, but obviously it's not great. So there's a few problems with it. One, the pancreas is super innervated. So there are long procedures if you want to do anything, but you can't have a pancreatectomy, sorry, I said that poorly, where you chop the whole thing out. You're chopping out part of the bottom of the stomach, you're chopping out the gallbladder, duodenum, and the whole pancreas. If you have this done, those patients, they instantly get one type of common disease. They become that disease type. What would that be? Can anyone think of that? If you take out the pancreas? What type of diabetic? Type 1, yes. So they instantly become type 1 diabetic because they are not making insulin, okay? There's another way of doing this called the Whipple procedure, or this long version of pylorus-preserving pancreatico-duodenectomy, where basically you're trying to preserve some part of the pancreas. So you're cutting off the head, but you're reserving this tail portion. The rest of it's pretty much the same, and then you have to reattach. So you're removing the pancreatic head, curve, and duodenum, but you're attaching the loop of jejunum to the cystic duct to drain bile. And basically, you're bypassing, the food's bypassing to the jejunum, okay? And so this is standard of care. You might see that on the boards, I don't know, but it's in first aid, so I included it. All right, I need to take this out, proprietary image. Are you guys familiar with the old McDonald's game at Monopoly? Yes, okay, you're right. No longer around. I used to love that. But anyway, what's happening here when you have some problem, you could have it for a few ways, but let's say you have a problem with the pancreas, or you're having pancreatitis, and so it's not working effectively, and you're ingesting various types of food. So if you have decreased pancreatic function, you're obviously having decreased lipase activity, okay? You also have decreased bile secretion production, and so you're not going to be able to handle these fats well, okay? So if you go to McDonald's, and when they were having this, and you ingest a ton of fat, a lot of that fat's going to pass through you because you're not acting on it, right? Your nanomachines aren't working. Other things, so these are causes of steatorrhea or oily stool. Cancer can also be there. Have you guys heard of orlistat? Yes, okay. So it's a lipase blocker. It can be used for weight reduction. I don't know if you'd consider that a huge efficacy or not. In 1 year, on average, you can lose 5 pounds. I think I talked to you about Olestra. Do you remember that in foundations? It is it's a fake lipid for taste that you can't digest. So I say it's the aspartame of fat. So I think Olay has the patent. It's out on the shelves, but if you can eat potato chips that are made with Olestra and you don't have the calories of fat, it's basically a carbon chain that you can't digest, but you can't digest it. So one of the side effects is I think like 8% of people that have that have oily stools because that oil is passing through them. And with all of those, if you can't work on the fat and the fat is going through you, that fat will bind other hydrophobic things like fat-soluble vitamins. And you have to worry about that, right? So, for example, on the Olestra label, they tell you, don't, I think they recommend that you basically not eat any, that you just sit there and house your potato chips and not eat other food with them. And if you were going to eat broccoli, you would have that 2 hours later so you can get fat-soluble vitamins out of the food later. Okay, this whole thing on the right, I'm not going to test you on. I believe you've seen that slide from me before. I'm just kind of reminding you about the cystic fibrosis aspect of this. So cyst fibrosis in the pancreas, the take home from cystic fibrosis was the CFTR is not working, chloride can't flow, and you end up having water, you end up with thick mucuses in a lot of places. And so likewise here, for the pancreas, you end up not being able to get your zymogens where they need to go, okay? So you have these thickened secretions, the pancreatic duct can get clogged, the pancreatic enzymes don't get to the intestine, and you can end up with chronic pancreatitis, which leads to EPI, exocrine pancreatic insufficiency. The treatment is replacement enzymes and supplementation with fat-soluble vitamins. Okay, pretty easy. But, and then I will say that this is a much rarer thing, but again, in first aid, so maybe on the boards, I wanted to fill in the picture that the second most likely way to get EPI, exocrine pancreatic insufficiency, is this Schwann-Diamond syndrome, SDS. So it's 5 per million people, autosomal recessive. And it is caused by almost everyone that has this mutation in SDS, the SDS gene, which is involved in ribosomal assembly. So you have decreased ribosomal assembly, which decrease your acinar cells function, decrease pancreatic enzymes. Okay, and we'll talk about we'll talk about kind of the duct a little bit more in a second. But this happens. This can't be kind of looked at via a history tree, family history, because this happens via gene duplication, which we've talked about multiple times. So at one point we had one SDS gene, then it was duplicated, we had 2, then the other one changed, so it became non-functional, which was fine because we still had the 1, but sometimes that non-functional one, since it's so similar, crosses over with the functional one and you lose both. So that's just going to happen randomly 5 out of 1 million times to people, okay? All right, so you have these, again, don't need to know any of these structures. You actually, people wonder what bile salts are, so I wanted to show you, but you can just totally put a black square over this thing on the right if you don't want to see it. So you have cholesterol, and then you have a corresponding bile salt. And the ratio of that is important, okay? If your ratio of cholesterol to bile salt increases, meaning you have more cholesterol than normal, you kind of want those to be equivalent, 1 to 1. If you have 3 cholesterol to 1 bile salt, I actually don't know what that ratio becomes a problem, but when it increases, your cholesterol hardens, right? And that creates these pebble-like solids, which are gallstones. And so it also leads to steatorrhea. So you have a deficiency in fat-soluble vitamins. So this ratio could get off for a few reasons. You could increase your cholesterol a ton, but more likely it is a problem with your bile, right? If you decrease your bile, that ratio is often the same way. Okay, autophagy. Okay. I have just circled what I really want you to know here. Again, and you've heard some of this story to me, but basically, autophagy is good, right? Autophagy is the controlled housekeeping of a cell, and mTOR is the hub that recognizes metabolism. So if you ingest food, mTOR is up, and basically we ingest too much food, mTOR is up too much. AMPK inhibits mTOR, AMPK is good. Okay, so how does this pathway actually work? And we're going to talk about how autophagy is related to pancreatitis. This creates these autophagosomes, which start as an isolation membrane. So you have these things in the cytosol that can be kind of thought of as organelles. So this, on the left here, this purple thing, it's actually a bilayer that goes around to another bilayer. And so it grows and kind of encompasses some cytosol, okay, when this is activated, and then you end up having an autophagosome, which goes on to fuse, forming an autolysosome. It fuses with the lysosome, okay? And so we'll talk about these snap-snare complexes, okay? So I want to go into this. So the general take-home here is I would imagine everything you will see on the boards would be mTOR is bad, right? So, and so if you inhibit mTOR, if you activate mTOR, it decreases autophagy, right? And so that's involved in a bunch of these bad things: cancer, diabetes, obesity, neurodegenerative diseases, inflammatory diseases, cardiac and muscle diseases. And if you increase autophagy, it's also associated with increased lifespan. I'm not going to test you on this, what I'm about to say. This one, I was debating if I should mention this. So if you want, shut your ears off for 45 seconds. Have I told you the one time mTOR might be good? No? Okay, so if you want, I won't test you on this in case this is in the boards. It could be very much in the clinic. The general take home is mTOR is generally bad and sirtuin is good. Remember sirtuin, circadian clock, blah, blah, blah. And those are on at different times. There's this one case you might see in the clinic where they're inhibiting sirtuin as a drug. So most of the things are there, you want sirtuin. And the one time they inhibit it, it seems to be that their testing is with cancers. So if you think of sirtuin as extending lifespan, the one time you don't want to extend lifespan is for the cancer cell. Does that make sense? So that you might come across something that seems like that's the opposite of everything Dr. Arbor said. If you have cancer, maybe you want to go in the opposite direction there, okay? All right, big FYI slide that autophagy is related in all these things and good. All right, so I'm going to go through these four pathways that a cell can die. I'm going to compare the middle two, intrinsic apoptosis and necrosis, okay? Before that, let's go through the two other ones quickly. You can have extrinsic apoptosis. That's where you're getting a signal from outside the cell that this cell should die. And so I colored that green because that should be good, that this cell is, that something's detecting maybe this cell's cancerous or infected with a virus. Anyway, normally that's good, and you signal through this death receptor, DR5, so that's good. Then there's failed autophagy. This is kind of a gradient. It depends. This could be good or bad, so it can't really be labeled either. But basically, if you have decreased nutrients, you can increase macroautophagy, and you're maintaining ATP production, and you have cell survival, but you can end up with this thing called failed autophagy. Don't worry about that because it's a mixed bag. I'm not going to teach about it because it's unclear. I want to go through these two things because it's nuanced, but you kind of need to get the difference. There's intrinsic apoptosis and necrosis. What I want you to see is that they come together at this spot where you have this open mitochondrial pore. And the take home is, if you open that a ton, it's bad, it goes this way. And if you open it a little, it's good. So what happens in intrinsic apoptosis? There's some intracellular stress. You have signaling through BCL2 that releases calcium, and you end up increasing this pore a little bit, and so your ATP goes down because you're reducing the gradient, okay, for your mitochondria, okay? And so this can be thought of maybe as like starvation mode or, right? And so that's good. If you open it up a ton, you completely lose your gradient, and so then you have necrosis. The cell's dying, it's kind of way too strong, okay? So, how does this all apply to what we're talking about? So if you have alcoholism that leads to pancreatitis, it decreases autophagy and increases necrosis. Especially, there's an oxidative and non-oxidative pathway for ethanol metabolism, and that happens via this open mitochondrial permeability transition pore, okay? Now, I know what I just said was really weird. Just focus on this opening pore, a little ATP going down versus a lot is bad and a little is good, okay? The picture is very complex. If you want to feel better about that, last year, this was the slide I showed students to explain that, and I'm not showing that to you or requiring that this year. It's complicated. There is a non-oxidative pathway in red and an oxidative pathway in green that's good, but basically the non-oxidative is bad. It opens this up too much, and ethanol also directly works on things. I will show through this cathepsin B thing in a second. So you don't need to know any of that. Just know opening up the membrane too much is bad. So you have this zymogen granule. You have a zymogen, right, that has to go somewhere. it can go into this autophagic vacuole, which then goes into the lysosome, which then can get degraded. Okay, that's a normal pancreas. I showed you that happening a few slides ago. Up here, okay? So that's normal. Okay, so what happens with ethanol? So first of all, here, this whole big picture is showing you a portion here, right? Here's your autophagic vacuole merging with the lysosome. So we're right at that step, okay? Ethanol directly inhibits this protein called LAMP2, which is involved in them fusing, okay? So that's bad because you're inhibiting that. Ethanol also inhibits cathepsin L, which is an inhibitor of trypsin. So you're inhibiting an inhibitor, right? So you're activating trypsin in a way. Okay. All right, so there's the take homes here. All right, so now let's talk about what's actually happening with the fusing and that space shuttle docking that I showed before. So you have these zymogen granules here. Every Z here is a zymogen, right? So this could be trypsinogen. And it has a V snare, which stands for vesicle snare. There's new words for this now, but I like V and T snare still. V snare for the vesicle. It goes up and binds to T snare and then releases the zymogens, okay? The zymogens go in the duct. Up here is your normal duct, and then you get to the enteropeptidase, which activates trypsinogen into trypsin, okay? So this is all good, okay? And how is that activated? It's by increased calcium in the cell. So you have these acidic granule compartments that have calcium that can be released, or you can release calcium through the ER, okay? And that signals for that to happen. So that's all good. But and the reason this happens is you have these, you can think of V snare and T snare as like the space shuttle and the space station, where they're persistent structures, right? So this vesicle can go up, dock, release it, come back down, get more zymogens, go up again. And so you have this in multiple things whenever you need to release stuff. I'll show you that in a second. Okay, so unfortunately, you can have things go awry, and ethanol can do one of those. So ethanol, or greatly increased CCK, basically can cause this vesicle to come down and release the zymogens back into the basolateral side, okay? And so you're releasing zymogens not into the duct, but backwards into the tissue, and you can cause necrosis of the tissue. Now, some great common questions I get that I don't know the answers to, that I've looked at and to me honestly seem unknown, are why would you have a T-snare on the basolateral side? I don't know. Maybe you're releasing something else that you want there and it's happening this way and whatever. I don't know the answer to that, but it can happen and it causes necrosis. All right, so you can see these porosomes, like here's an electron micrograph of one, and here's their structure, blah, blah, blah. The take home that I want you to get here is this is not just in the pancreas. So here's the pancreas and here's neurons, and they both have these porosomes. In the pancreas, they're super big, 150 nanometers, and in the neurons, they're 12 nanometers. And then how long do you dock there and release stuff? In the pancreas, they can sit there minutes and release a ton, and in the neurons, it can last milliseconds, okay? So why do I bring this up? This is definitely not going to be on the boards, but during your career, I could imagine drugs being created that target the porosome, and if they target it generically, you might be trying to target it for pancreatitis, but it could affect the brain if it's targeting the porosomes there too, right? It depends what part you're targeting. If you're targeting a generic T-snare that's expressed in both tissues, then you have to worry about that. The V snare binding to the T snare is actin dependent. So if you have anything affecting that actin transportation, it will affect this. So for example, this cytochalasin B is a fungal toxin, and it inhibits the actin polymerization, and it inhibits cell secretion because of that, okay? So the actin is kind of your highway where you're bringing vesicles to release stuff, and if you put a lot of potholes in your highway, you can't get the vesicle to the membrane to release it. All right, so the quick thing about omega fatty acids, omega-6s are bad. They they get converted to arachidonic acid, which get converted to eicosanoids, which cause inflammation, okay? And they're upregulated in a lot of cancers. And this is, this conversion occurs from COX and LOX, and we have COX and LOX inhibitors. So there's all these trials are FYI, but I wanted to show you the data on these inhibitors for pancreatic cancer don't seem to have great outcomes. So the red here is no significant outcome, no significant, and then a few good ones. So maybe the magnitude of that inflammation, limiting that is not enough of an effect, right? All right, so how do you treat pancreatitis? Basically, you can treat it with enzymes. I find this amazing that we do this. So pancreatic enzyme replacement therapy, you have amylase, lipase, and chymotrypsin, right? Amylase works on the carbs, lipase on the fats, chymotrypsin on the proteins. All of these are made from a pig pancreas. So I just find it amazing that we can take animals, use their body as nanomachine factories for us, and then take in those enzymes, and that is a functional therapy, and it works great. There's a bunch of types here, Creon, Zenpep, pancreas. The only one I'd point out is Viacase. I don't know if that is still used often, but it is an uncoated version, and the problem with that was you needed to take cimetidine with Viacase because it's an H2 receptor antagonist that inhibits stomach acid production. So you basically didn't want the enzymes to be degraded in the stomach, so you kind of raised the pH of the stomach, but the problem was you would then have opportunistic infection because bugs can get through because you're getting rid of the harsh acid environment to handle them, okay? So you can also have replacement therapy of bile acids, and so that can dissolve gallstones. It can be used preemptively for rapid weight loss too. I don't know if people actually do this, but if someone's like an obese patient against your guidance is going on like a crash diet for 6 months, they're more likely to develop gallstones there. And so you could proactively have them taking bile acid supplement. All right, so this would be the table to study to kind of get at the, in the stem of the question, like what this is highlighting towards acute or chronic pancreatitis, right? Gallstones for acute, alcohol abuse for chronic. You have for pathophysiology, I'll just go down for acute, you have activation of pancreatic zymogens and autodigestion of the pancreas. So that is necrosis, right? You're digesting your own tissue. You have pain radiating from the back, shock and fever. The gold standard is a CT scan. That's the most accurate. You can also detect amylase in the blood. And then the treatment are IV fluids, bowel rest, and antibiotics. This is a really expensive thing to treat because there's a lot of inpatient care, right? So there's a lot of people, a fair number of people have it, 200,000 per year, and that costs us over $2.5 billion per year, over 10,000 each. For chronic, you have replacement of deficient enzymes for chronic pancreatitis. All right, that is it. Any questions? All right, if you have personal ones, come on up.